Hello, everyone. My name is Anthony Covarrubias. I'm an assistant professor at UCLA in the Microbiology, Immunology, and Molecular Genetics department. I'm really happy to join you here in the Metabolic Physiology seminar series, and I want to thank the organizers for giving me this opportunity to share my work, in particular, Dr. Phil White. So today I'm going to tell you a story that a lot of it was actually done during my postdoctoral training at the Buck Institute in UCSF Gladstone Institute when I was a postdoc with Eric Verdin. And this was recently published a few months ago. So I'm really excited to share this story with you. And it's looking at the role of immunometabolic dysregulation in macrophages during aging, and really aging at the crossroads of inflammation and metabolism. So we classically think of our immune cells as cells that protect us from pathogens like viruses and bacteria, which they do a really good job of doing. But over the last decade or so, there's been the field of immunometabolism emerging. And that's really the appreciation that immune cells do a lot more than just protect us from pathogens, but play a really important role in tissue homeostasis and metabolism. For example, cells like the microglia, which are macrophage-like cells, which are seated during development in the brain, play a really important role in brain tissue homeostasis and, for example, for pruning synapses. We also know other immune cells reside in high abundance in other metabolic tissues, including the adipose tissue and liver. So in the liver, for example, you have Kupffer cells or macrophages, which interact with hepatocytes in the liver to regulate both lipid and glucose homeostasis. And the same thing happens in the fat tissue where adipose tissue macrophages or ATMs regulate lots of the biology of the adipocytes in the adipose tissue itself. So a lot of my training actually has been in the field of nutrient sensing in the immune system to try to understand, do immune cells that reside in these metabolic tissues where there's lots of flux of different nutrients occurring, can they directly sense nutrients in their microenvironment, or do they sense it indirectly? So that was a major question I wanted to ask during my PhD training. And I was fortunate to work with Tiffany Hong at the Harvard School of Public Health. And to answer this question, we actually identified the role of the mTOR-AKT pathway in regulating the nutrient sensing in macrophages. So the immune cells, what we found is can directly sense cell autonomously nutrients in their microenvironment. And the nutrients actually play a really important role in the activation state of the immune cells in these metabolic tissues. For example, during cases where you're in a fasting state and have lower nutrient levels, the immune cells like macrophages can dial down their activation state to conserve energy. Conversely, when you're in a postprandial or fed state, when nutrients are higher, we now know that the macrophages can sense this high level of nutrients directly and then increase their activation and gene expression of pro-inflammatory cytokines, as well as other factors that regulate metabolism. And we also know that under conditions where there's chronic excessive nutrients, which occurs during obesity, that these nutrient sensing pathways can be overactivated, leading to aberrant dysfunction of the immune system. So that's kind of highlighted here in this box from this review, from this image. And essentially what it's showing is that in conditions where you have nutrient excess, which occurs in obesity, you generally get nutrient energy stress, as I mentioned, because of overactivation of nutrient sensing pathways, which leads to aberrant dysfunction of immune cells or meta-inflammation. Essentially inflammation and immunometabolic dysfunction, which actually is a driver of lots of these diseases. For example, inflammation associated with immune cells under obese conditions can exacerbate and promote insulin resistance. And we now know that this nutrient energy stress is also a key driver of other diseases, including cancer, neurodegeneration, and asthma. And it's actually becoming more and more identified as a causal role in aging. So inflammation is emerging as a key driver of aging-related diseases, and that's kind of what I would like to tell you a story about today. So a lot of my background, both my training and my future work in my laboratory at UCLA, is really looking at the intersection of inflammation and metabolism and how immunometabolism regulates different disease states, including aging, but as well as cancer and metabolic disease. So I previously mentioned in introduction that metabolites have a profound influence on immune cell function and regulate inflammation. And today I'm going to tell you a little bit of the reverse relationship here, and that's how inflammation can regulate metabolites. One of the major metabolites I'm really interested in is actually the metabolite NAD. So as shown here, here's the NAD, or the dinucleotide, nicotinamide adenine dinucleotide. And this molecule is actually essential for life. So all of us probably remember NAD from our biochemistry classes. It's actually found in every cell in our body and all living organisms, and actually found in high abundance in different organelles as well. And NAD metabolism is actually very complicated. If you're interested, we wrote a nice review that just came out a couple months ago in Nature Reviews Molecular Cell Biology, where we go through all these very complicated pathways in depth. But for today's talk, I just want to make the key point that NAD is a really critical electron acceptor and donor, and is essential for lots of metabolic reactions, including the TCA cycle in the mitochondria, as well as glycolysis. It also acts as a key coenzyme for hundreds of different enzymes, and a lot of these different enzymes have still yet to be discovered, and it's a really exciting area to study NAD metabolism and NAD biology. But two of the more important ones that have been identified in aging are sirtuins and PARPs. So these are stress response proteins that play an important role in promoting healthy aging. And what's really interesting about the NAD levels in aging is that it's actually been observed that, at least in rodents and potentially in humans, that as mice age, generally a young mouse has a really full replete NAD level, as shown here in this kind of schematic. And as the mouse ages approximately 2 to 3 years, which is natural lifespan, NAD levels have been found to decrease roughly 50%. And that's kind of demonstrated here in this nice review from Shin and Mai a few years ago. And what they showed in this review is that if you look at different tissues, including the muscle, brain, fat tissue, as well as liver and other tissues they looked at, that multiple labs around the world have consistently saw a decrease of average about 50% in all these tissues during the course of aging in mice. And of course, this decline in NAD is important for looking at, because we now know that it impacts a lot of different cellular processes. And some of them are included here, but and include, for example, epigenetics, which we know gets dysregulated during aging and can be reversed by supplementing NAD precursors, which activate sirtuins. Other critical cellular processes include circadian control of gene expression, as shown here, as well as the control of DNA repair and chromosome stability. And we know this because acute DNA damage activates a DNA repair response, which is actually dependent on lots of NAD-dependent enzymes, including sirtuins and PARPs. So as NAD levels decline, DNA repair is thought to become more and more inefficient. And of course, this impacts the health of aging mice. For example, a decrease in NAD has been linked to increase in cancer, as well as increased cellular senescence. A decrease in NAD has been linked to metabolic dysfunction, as well as a general physiological decline, which ultimately has an impact on the health span and lifespan of rodents and potentially humans. So there's been a lot of work actually to try to restore NAD levels to more youthful levels in aging mice. And that's mostly been done by using NAD precursors such as nicotinamide riboside or NMN, which have actually been shown to restore NAD levels in aging mice and actually reverse some of these aging-related pathologies. However, when I started this project, there was still an open question in the NAD and aging field, and that was that we didn't really fully understand why NAD levels were declining in the first place. And that was something I really wanted to answer, because I thought it was very fundamental to try to understand better the biology of aging, but as well as how we could make NAD levels replete more efficiently. And essentially what I wanted to do is find a way to plug the sink and figure out why NAD levels are being degraded during the aging process. And there's some different hypotheses that were occurring in the aging field as to why there may be a decrease of NAD during aging. So as shown here in this kind of very simple schematic, this is a molecule of NAD. And NAD can be consumed by lots of different enzymes. Some of them included here are sirtuins, PARPs, as well as CD38. And what's common amongst all of these different enzymes is that they make a byproduct once the NAD becomes consumed, which is nicotinamide. And NAD is so critical for life that our cells, even though they consume a lot of it, always have found a way to keep NAD levels sustained. And that's mostly via the NAD salvage pathway. There's multiple enzymes in the pathway, but the most important one is thought to be NMPT. And this pathway can convert nicotinamide back to NAD or salvage the byproduct back to NAD. So it's thought that perhaps during aging, because you have more stress, for example, metabolic stress or DNA damage, that you have more stress response proteins being activated like sirtuins and PARPs, and there's just a greater demand for NAD. So perhaps during the aging, there's this, the demand for NAD increases. And that's one reason why there may be a decrease in NAD in aging. The other hypothesis is perhaps that maybe there's a defect in the NAD salvage pathway, which makes the recycling of nicotinamide to NAD just more inefficient. Or perhaps, and more likely, it's really a combination of these effects, that you have greater demand and the inability to convert nicotinamide to NAD during aging. So this is a question that lots of people in the aging field have been trying to answer over the past few years. And around the beginning of my postdoc, there was a really nice paper that came out from Eduardo Chini's lab at the Mayo Clinic, and they wanted to investigate the expression of some of these different NAD-consuming enzymes, as well as enzymes in the NAD salvage pathway in aging tissues. So what they did is they took lots of different tissues from aging mice, including the muscle and fat and liver, and then measured the expression of all these enzymes. And here's the data from the fat. And so what you can see is they looked at mice that were roughly 5 months of age to 32 months of age, so really spanning the whole lifespan of the mouse. And what they found is that when they looked at the expression of NAPT, the critical enzyme in the NAD salvage pathway, that there was really no difference in its expression. So suggesting that perhaps maybe decrease in NAPT wasn't a major causal factor in declining NAD. They also looked at expression of SIRT1, and also similarly saw no effect. Interestingly, PARP1 seems to decline with aging, suggesting PARP1 is not a major consumer. But when they looked at CD38, they saw a nice and linear increase in CD38 expression with aging, suggesting that CD38 may be a major consumer of NAD that increases with the aging in multiple different tissues they looked at. So they did a really nice experiment where they took wild-type mice and then measured NAD levels in abdominal fat as well as other tissues during the aging process. And as expected, NAD levels declined roughly 50% in these tissues during the aging process. However, when they aged CD38 knockout mice, they saw that these mice were actually fully protected from NAD decline during aging, which is really strong evidence that CD38 perhaps is the major consumer of NAD and is really just degrading NAD. And of course, you're probably wondering what CD38 is. So CD38 is actually a really interesting molecule or protein that's expressed mostly on activated immune cells, but also in some other cell types, including endothelial cells as well. And this molecule it's thought to be an ectoenzyme with its enzymatic domain facing outwards and can consume NAD and essentially cleave it in half to nicotinamide and ADP ribose. We also now know that CD38 could also be found inside the cell as well, not just on the cell surface. And something that they didn't show in this paper, so this paper is really interesting, but there were still some outstanding questions left to be answered. And one of them was that because they were only looking at whole cell tissue lysates, we didn't really fully understand what cell types were expressing CD38 during aging, and we also didn't understand what was driving CD38 expression. So these are some of the questions I wanted to answer during my postdoc. And because CD38 is actually found predominantly on activated immune cells, I thought perhaps there was a link to inflammation driving CD38 expression. So that was kind of the hypothesis starting this project. Is aging-related inflammation a key driver of CD38 expression and NAD degradation during aging? So to answer this question, what cells express CD38 in aged tissues, I looked at the abdominal fat, mostly because in the Eduardo Chini paper, they saw the biggest increase in CD38 aging in abdominal fat. So I wanted to focus on this tissue. And also I wanted to repeat the study just to verify whether I could replicate these studies. And indeed, I was able to. So shown here, if we take abdominal fat from young and old mice, in this case, 3 months and 24-month-old mice, we could see that as the mice age, we see roughly a 50% decrease in NAD levels. And just as the previous paper suggested, when you age CD38 knockout mice, they have a much more significant amount of NAD compared to their wild type companions shown here. So suggesting that CD38 is indeed the major consumer of NAD. And then what I wanted to do next was then take the abdominal fat or visceral fat from the mice, from both old and young, and then separate the cells so we could digest the tissue. And what's really nice about abdominal fat is that the lipid-laden adipocytes will float to the top when you spin down the digested tissue. And a lot of the different immune cells will spin down to the bottom in the fraction called the stromal vascular fraction, or SVF. And then I could take this stromal vascular fraction as well as the adipocytes and analyze them for CD38 expression. So that's what I did by flow cytometry. We didn't find any expression of CD38 really in the adipocytes, and so we focused more on the SVF fraction. So I stained these different cells in SVF with different markers to identify them, as well as CD38 antibodies. And we really didn't find any cell type that was changing its expression of CD38 besides the macrophage, actually. And what's really interesting when we saw this, when we started analyzing macrophages, really, one of the first things we noticed is that the resident population of macrophages was actually increasing during the aging process. So as shown here, this is a young 2-month-old mouse. And as it ages to 25 months, we can see there's already almost a doubling or tripling of the amount of macrophages found in the fat tissue of old versus young mice. And then what we saw is that a lot of these resident macrophages actually are have very high expression of CD38. So as shown here, we see that CD38 is generally not highly expressed in younger mice, but as they age, the amount of CD38 positive macrophages increases. And this is kind of this quantification of the overall abundance of macrophages in young and old tissues, which I thought was actually very striking and interesting. So in this case, we take male and female mice and stain for the macrophage marker F480. You can see that there's not as many macrophages in young tissue, but as the mice age, the abundance of F480 staining becomes much more obvious. So which is very consistent with our flow cytometry results. And what we wanted to then do next is then ask, do these CD38 positive macrophages consume more NAD? So to do this, I took a more in vitro approach. And macrophages are very interesting because they can take on distinct different subtypes, which includes the anti-inflammatory like M2 macrophage, which is thought to be kind of anti-inflammatory macrophage, or more like a kind of a macrophage that regulates tissue homeostasis. And these macrophages can be activated by the Th2 cytokines IL-4 and IL-13 are thought to play a really important role in metabolism, as well as wound healing and in some cases fibrosis. Whereas an M1 macrophage is thought to be more the pro-inflammatory macrophage, which responds to different pathogen-associated molecular patterns like LPS, and plays a role in fighting pathogens, but an M1-like macrophage are thought to be a key driver of lots of chronic inflammation, which occurs, for example, in aging, but as well as in other cases, for example, in the case of gout here. So what I wanted to do was to polarize macrophages to the M2 state as well as to the M1 state, and then measure different expression, the expression of different NAD-consuming enzymes, including CD38. That's what we did here in this first experiment. And we polarized macrophages from 4 to 24 hours to the M2 or anti-inflammatory macrophage, as well as the pro-inflammatory M1 macrophage. And what we found was actually very striking and very clear was that it was really only the pro-inflammatory M1 macrophage shown here that increased its expression of CD38, as well as its homologue CD157. And when we looked at different sirtuins and PARPs, we saw that these enzymes really didn't change under macrophage polarization conditions. And what we wanted to do next was to then see if these M1 macrophages were consuming more NAD. And to test this, we actually use a really interesting molecule called epsilon NAD. So this is a NAD molecule that when it becomes cleaved, it can fluoresce. So the byproduct fluoresces. So we could use this as a proxy for NADase activity. So that's what we did here. Using both host cells as well as host cell lysates, we can take M1 macrophages as well as M2 or M0, which are naive macrophages, and then measure the amount of NADase activity both at the host cell level and at the tissue, the cell lysate level. And as you see, it's really only the M1 macrophage that has really high rates of NADase activity, as shown here, whereas the M2 and M0 macrophage have very little basal NADase activity. So NADase activity seems to be really a hallmark of pro-inflammatory macrophages. Another way we can measure NAD degradation is actually via metabolomics. So as I mentioned earlier in the previous slides, that NAD-consuming enzymes make nicotinamide as a byproduct from once NAD is cleaved. So we could use metabolomics and mass spec to look at the byproducts of and different metabolites related to NAD in both the M1 and M2 macrophages. And that's what we did here. So as shown here, this is measuring nicotinamide, which is the byproduct of NAD consumption. You can see really it's only the M1 macrophage that has a large increase in production of nicotinamide, which is consistent with these M1 macrophages turning over lots of NAD. And we don't really see this in the M2 macrophage as shown here. So to summarize this slide, what I'm showing you is that M1 macrophages seem to be characterized by really a high rate of NAD activity and consumption of NAD. And what we wanted to do next was see whether this really high rate of NAD activity found in pro-inflammatory M1 macrophages was CD38 dependent. So to test this, we utilize CD38 knockout macrophages. So as shown here, if you compare it to a wild type macrophage, it's really just a wild type M1 macrophage that express high levels of CD38. And of course, our CD38 knockouts are knockouts that they don't express CD38. We also see that, as we previously mentioned by qPCR, a little bit of CD157 or CD157 is also co-expressed with CD38, both in wild type and CD38 knockouts. But interestingly, when we looked at PARP expression as well as different sirtuins and NAPT, none of these other NAD consuming enzymes as well as NAPT change during macrophage polarization. It's really just the CD38 and its homologue that seem to be increasing under conditions or pro-inflammatory conditions. And we repeated the NAD assay again, now using wild type macrophage, which as expected increased lots of, have really high rates of NAD activity. But now you can see here, if you compare CD38 knockout M1 macrophage, it looks very similar to our M2 or M0 macrophages. So you almost completely lose this high rate of NAD activity by knocking out CD38. I don't have time to show you here, but we also use double knockouts as well as CD157 knockouts. And we actually found that CD157 doesn't seem to be responsible for this high rate of NAD activity. It seems to be solely dependent on CD38. So this is quantified here. So you can see that 95% of NAD activity in M1 macrophage seems to be CD38 dependent. And CD38 seems to be exclusively expressed by pro-inflammatory macrophages. So since we were seeing lots of CD38 expression in vivo in aging macrophages, and CD38 expression seems to be associated with inflammation, we wanted to test whether we were seeing evidence of inflammation in aged adipose tissue during aging. So here I took young and old mice, in this case 6 and 25-month-old mice, and then measured some canonical pro-inflammatory cytokines. So as you can see here, we see a nice increase in inflammatory cytokines like IL-6 and IL-1 beta, which are major cytokines implicated in inflammation and aging. Consistent with our flow cytometry in previous results, in vivo, we saw there was a nice and significant increase in CD38 as well. But what's interesting is we also saw an increase in CD68, which is a macrophage marker. So this increase in CD38 seems to be associated with increased inflammation and increased macrophage abundance, which is, as I mentioned, consistent with the results we found from flow cytometry. We also looked at M2 macrophage genes. So these include, for example, genes like arginase and FIZ1 and MGL2. And we saw that despite there being a lot more macrophage abundance in fat tissue, we didn't see any increase in the expression of these M2 markers. And in fact, a lot of them seem to be kind of trending downwards in aged tissue versus young, which suggests that perhaps there's kind of a switch from an M2 macrophage to an M1-like macrophage during aging. And to just to validate some of our qPCR studies, we wanted to look at kind of the protein level by Western blot at some of these different markers. So what I included here is this a Western blot of young and old adipose tissue, and then looking at the marker for CD68. So you can see it's a very striking and black and white kind of result where you see that there's much more CD68 expression, which is consistent with there just being a lot more macrophages in old fat. And we also saw that there was also activation of the inflammasome, which is an immune complex that regulates the activation of IL-1 beta as well as IL-18, and it's a pro-inflammatory kind of pathway found in a lot of innate immune cells. And we can see that this pathway is activated because when the inflammasome becomes activated, it self-cleaves itself, and we could find a 20 kilodalton version of caspase-1. So much more activation of the inflammasome in old versus young mice, which is very consistent with there just being a lot more inflammation in old versus young mice. Something else that we found is that there's a lot more evidence emerging that senescent cells are maybe perhaps a key source of inflammation in aged tissue. So we also wanted to look at expression of senescent cell markers as well. And we also, and consistent with previous studies, we saw there was a very significant increase in senescent cell markers, including P16 as well as P21. Another hallmark of senescent cells is that they seem to accumulate the enzyme beta-gal in their lysosomes. So you can use the substrate X-gal and then see if the, which will be cleaved by beta-gal and turn blue. So you could treat old tissues, for example, fat tissue with the X-gal substrate, and if the cells turn blue, that's evidence of senescent cell burden. And that's demonstrated here. So we took young and old fat and then added the X-gal substrate, and you can really visibly see the old tissue turning really dark blue compared to young tissue, which is really strong evidence that there's a lot more senescent cells accumulating in old tissues. And this is just looking at IF of P21. So this is the marker here, senescent cell marker P21. And you can see that in old tissues, we're seeing lots more staining in fat tissue of P21 versus young tissue. So further evidence that senescent cells may be also a key source of inflammation in aged tissues. So what I'm showing you in this slide is that during the aging, adipose NAD decline is associated with chronic inflammation and increased numbers of senescent cells. And what we wanted to do next was then ask this question: is there a link between senescent cell burden and aging and expression of CD38? So just if you're not familiar with senescent cells, these are cells that have generally undergone some type of stress, for example, DNA damage that causes them to undergo cell cycle arrest. And there are some benefits to this gene program, including its benefit as an anti-cancer program. So you don't want cells with DNA damage to keep proliferating and potentially turn into cancer. However, the problem is that once these cells reach a critical amount, they actually they secrete lots of pro-inflammatory cytokines, including cytokines and chemokines and growth factors, and which actually cause a lot of tissue dysfunction and inflammation. So what we wanted to do is to utilize young mice and inject them with a drug that can cause senescent cells to increase, and see whether we can see whether this impacts CD38 expression in macrophages. So in this case, we use a molecule called doxorubicin, which will cause DNA damage and the accumulation of senescent cells. And what we found is when we looked at mice injected with PBS as a control or doxorubicin, was that as expected, we see a nice increase in senescent cell burden. So we see really high expression of these senescent cell markers, P16 and P21. Just like old mice, we see a nice and significant increase in expression of inflammatory cytokines like IL-6 and IL-1 beta, and also an increase in the senescent cell, or the macrophage marker CD38, as well as an increase in CD38. So really kind of mimicking what we see in young versus old mice. And what we wanted to do next was to see whether this increase in CD38 expression was linked to macrophages. Just like we saw in old mice, we see that the total amount of macrophages in the fat tissue of these doxorubicin treated mice actually increases. And we saw that these macrophages were actually CD38 positive. So we could replicate the results in aging by treating mice with doxorubicin and really finding a link between senescent cell burden as a causal regulator of CD38 expression in macrophages. But we didn't really know whether this is an indirect or direct activation. And that was something that we wanted to answer next. So to answer this question, we took fibroblasts and made them senescent cells. So in this case, we could treat fibroblasts with doxorubicin, as well as irradiate them and cause acute DNA damage. Fibroblasts will undergo the senescent cell program, and by day 10, they're secreting lots of SAS. So on day 10, we collected the SAS, or conditioned media. We filtered it and got rid of the cell and decelled debris, and took this fraction on top, which contains the SAS components like cytokines, and put it on macrophages. And then we waited 24 hours and then measured gene expression. And this is what we found. So we looked at the expression of lots of different NAD-consuming enzymes. And just like we saw kind of in one macrophages, we saw that the conditioned media from senescent cells, but not control fibroblasts, was actually able to cause a nice increase in CD38 expression and similar increase in CD157, it's homolog, but not PARPs or sirtuins. So, what this data suggests is that it's really the SAS factors that are being secreted by these senescent cells that are perhaps directly activating macrophages to kind of a pro-inflammatory M-like state, which is, as we shown, is characterized by high expression of CD38. And what we wanted to do was, a lot of the studies were, to this point, were only done really looking at adipose tissue. And we wanted to see whether senescent cells can affect CD38 expression in other tissues. So we were very fortunate to collaborate with the Chan Zuckerberg Biohub and the Tabelmeyer Senes Consortium to utilize their really interesting study where they did lots of different single-cell RNA-seq analysis of multiple different tissues in young and old mice. And here I'm just showing you the liver data. So this is a single-cell RNA-seq analysis or t-SNE plot from mice aged from 1 to 30 months. And as shown here, you can annotate the cells by cell type. So this includes, for example, hepatocytes. These magenta cells here are the Kupffer cells, or the resident macrophages of the liver. And here is endothelial cells and other immune cell subsets. And we wanted to ask what cells within the liver express CD38. So that's shown here. So it's really two major sources of CD38 in the liver, and that includes the Kupffer cells or macrophages, as well as the endothelial cells. And we can also annotate these cells by the age in the mouse. And that's shown here. And what was very remarkable, actually, and I thought was very interesting, is when we annotated, looked at the annotation by age, we could see that unlike the endothelial cells, which will cluster, all the mice from different ages seem to cluster into one group, Kupffer cells or macrophages seem to cluster by age. So as you can see here in purple, these are the Kupffer cells from a mouse that's 1 month old. These are the Kupffer cells from a mouse at 18 months and then at 30 months. And what this data suggests is that Kupffer cells are undergoing lots of significant changes in gene transcription during aging. And what we wanted to ask was, in addition to CD38, was there other NAD-consuming enzymes or enzymes that regulate NAD? Are they changing in Kupffer cells during aging, as well as the expression of pro-inflammatory cytokines? So that's kind of demonstrated here in this dot plot, as well as this heat map, which are essentially showing the same data. But I want you to kind of focus here. So this is the expression of CD38 in Kupffer cells from mice that are 1 to 30 months, as shown here. And what we saw was there was not only an increase in the overall amount of abundance of macrophages expressing CD38, but that the overall expression also increases. And that tends to occur around 24 to 30 months of age. And what's really interesting about this result is that this seems to be the same time where we're seeing the expression of, or the accumulation of senescent cell burden in the liver. So the timing seems to be consistent with potentially senescent cells causing this increase in CD38 in aging Kupffer cells, as shown here. And what's also interesting is that consistent with CD38 being the enzyme that's increased in pro-inflammatory M1 like macrophages, when we looked at the expression of inflammatory cytokines in these Kupffer cells, we saw, for example, that IL-1 beta nicely increased very dramatically, actually, from 1 to 30 months. So you can see there's a very striking increase in the expression of this cytokine during the aging process, ultimately becoming very high by 24 to 30 months, which is consistent with these Kupffer cells being pro-inflammatory M1 like macrophages. And of course, we wanted to validate some of these results. So we did some IF of the livers of young and old mice, in this case, wild type mice. And here's a knockout. And we could then measure macrophages or Kupffer cells with the marker F480. So as you can see here, the overall amount of F480 seems to increase in old livers, similar to what we saw in adipose tissue. But not only the F480, but also the amount of CD38 seems to increase as well. So as you can see here in green, there's a much higher expression of CD38 in old livers versus young. And just to show that our antibody is very specific for CD38, we don't see any staining of CD38 in our knockout livers. And what this data collectively shows you, both the single cell as well as our IF, is that aging Kupffer cells seems to express higher amounts of inflammatory cytokines, So they're essentially in one like macrophages, which are characterized by high expression of CD38. And we could also quantify these IF images and try to see if we could see more co-localization of CD38 with F480, just to kind of validate the sequence RNA-seq results. And as shown here, we can see that old mice tend to have more CD38 positive Kupffer cells, or at least co-localization of the two markers compared to young mice, which is consistent with Kupffer cells as having higher expression of CD38. And what we wanted to then ask was, are these macrophages or CD38 positive macrophages in the liver, as well as in the fat, affecting systemic NAD levels? So to answer this question, we use both a chronic dose of LPS, which will cause macrophages to undergo an M1 polarization in vivo, as well as an acute dose. I'm just showing you the data from the acute dose, but we saw similar results in both chronic and acute LPS dosage. In this case, we injected both wild type and CD38 knockout with LPS, which causes a lot of inflammation. And we expect to see an increase in CD38 in these tissues, which we did. And then what we wanted to do was then measure by metabolomics the metabolites, including NAD as well as nicotinamide. And what we saw in fat tissue was really interesting. So in gray is the wild type mice. So this is PBS and LPS injected. So you can see after 12 hours, the wild type mice are actually have about a 50% decrease in their of NAD levels in the fat tissue. Similar, we saw a decrease in AADP, but interestingly, the CD38 knockout mice that were injected with LPS, we didn't really see a significant decrease compared to PBS of both NAD and NADP. The other molecule that we looked at was nicotinamide, and this was actually some really surprising results. And one is that the first one that's very obvious is that when you knock out CD38, that CD38 seems to be the major source of nicotinamide in aging tissue. So you can see that almost 90% of the nicotinamide is decreased in CD38 knockouts versus wild type. The other result that was surprising is that unlike in M1 macrophages in vitro, when we treat them with LPS and we see an increase in nicotinamide as a result of increased CD38 expression, we didn't see a significant increase in NAM in vivo tissues. And I'll get to the reason why I think that's the case after I show you the liver data. So here's the liver data, and we saw that there was an LPS injected mice that similar to the fat tissue, you see almost a 50% decrease in NAD levels in the fat tissue in wild type mice. But we also saw a pretty significant decrease in CD38 knockout mice as well. So NAD seems to be decreasing in the livers of both wild type and CD38 mice. And we actually found that in CD38 knockout, or in LPS injected mice, a lot of NAD in the liver is actually derived from tryptophan, or the novel synthesis of NAD. And this actually gets inhibited during LPS treatment. So inflammation actually causes a lot of tryptophan to be diverted away from the de novo pathway. And tryptophan seems to be converted to kynurenine. So it's no longer be kind of going down the de novo pathway. It seems to be kind of shifting to production of this other metabolite. And similar to fat tissue here, we saw that we didn't see an increase in nicotinamide levels as expected in LPS treated mice. And and this is actually very surprising, right? Because although we're seeing a decrease in NAD levels upon LPS treatment, we don't see the expected nicotinamide levels. And we thought perhaps that nicotinamide was going somewhere else. So what I wanted to do was look at another alternative pathway for nicotinamide, and that's actually this. It could actually be methylated by this enzyme NNMT. So we indeed found that upon LPS treatment in mice, that LPS actually causes a significant increase in expression of this enzyme. And then when we looked by metabolomics, we saw that in the liver as well as in the fat tissue, that we were detecting a significant increase in methylated nicotinamide, as well as its downstream products, 2-PY and 4-PY. But we didn't see this actually in CD38 knockout mice. You can see here that LPS treatment doesn't cause this increase in methylnicotinamide, only in the wild type mice. And what this data suggests really is that upon inflammation, you have an increase in CD38, which is actually consuming NAD and making nicotinamide. But instead of nicotinamide then being converted to NAD, it actually seems to be methylated into these metabolites, methyl nicotinamide, which can then be secreted in the urine. So essentially what I'm showing you is that CD38 and NMT are working in tandem together to divert this byproduct of NAD consumption, nicotinamide, away from the salvage pathway, which is contributing to the decrease of NAD levels during inflammation. So that's essentially the story that we found. And just to kind of rehash what I showed you today, is that during aging, you have greater inflammation as a result of greater senescent cell burden in aging tissues. And what we showed is that these senescent cells are secreting lots of different inflammatory cytokines in what we call the senescence-associated secretory phenotype, or SASP, and that these SASP molecules are activating macrophages in their microenvironment to a pro-inflammatory M1-like state, which is characterized by high expression of CD38, which is further contributing to inflammation and reduction of NAD. And we show that this is something that doesn't just occur during conditions of chronic inflammation, such as in aging, but also even during acute inflammation. For example, I just showed you the data where we treated mice with LPS. So it seems to be something that's common under conditions of inflammation. And what I also showed you earlier is that CD38 seems to be the primary NAD-consuming enzyme in M1 macrophages. We also showed that, really highlighting this novel link, and really for the first time showing that senescent cell burden is linked to NAD decline via CD38 and M1 macrophages. And lastly, I showed you with the very last slide that excess nicotinamide derived from CD38 is being methylated by this enzyme NNMT and being diverted away from the NAD salvage pathway. And I'm really excited about this model because it gives us actually multiple nodes where we can use for therapeutic targets. One is that we could just target the senescent cell burden. For example, there's been a lot of studies now using senolytics, the xenomorphs, which I believe based on our model should have an impact on NAD metabolism, something I would like to test. Another is we could just target the macrophages directly. So we could either block their different inflammatory pathways that are regulating CD38, or just target CD38 directly with different small molecules as well, which should be able to hopefully restore NAD levels. And has actually recently been shown in a recent paper from Eduardo Cini's lab in the Mayo Clinic. So I think all these different combinations can target kind of this relationship between senescent cells and macrophages to help restore NAD levels in aging, and hopefully improve aging and health span. So this prior work was actually my recent postdoctoral work that I published just a few months ago, but I now have a new position here at UCLA in the MIMG department, and I hope to continue to study the intersection of inflammation and metabolism in the context of different disease states. I hope that kind of was a nice kind of preview of some of the projects that my lab will be working on. And as a future direction, what I hope to do is to continue to study how inflammation and metabolism are integrated to control immune cell function. So really focusing on really fundamental pathways and mechanisms in immunometabolism and how they control immune cell activation. And I hope to continue to explore how different nutrients and metabolites are sensed and how they regulate immune cell function, as well as continue to investigate NAD metabolism as a therapeutic target in different disease states. And lastly, I hope to continue to kind of explore this novel crosstalk that I found between immune cells and senescent cells, and try to understand what are some of the mechanisms how senescent cells are regulating immune cell function. And I believe this is also a bi-directional relationship, and I believe immune cells also may be impacting senescent cell biology as well. So I really hope to kind of study this crosstalk and reveal some really cool and novel biology in my laboratory. And I think all of these projects collectively will have a major impact and play a role in different diseases that we're interested in studying here at UCLA, including infectious disease, metabolic disease, cancer, and even neurodegeneration. So with that, I'd just like to thank everyone for tuning in, and I hope to take some of your questions at the end of this talk. And of course, I want to thank all my collaborators who helped me with this project. And I first and foremost want to thank my postdoctoral mentor, Eric Verdin, who gave me lots of resources and support to pursue this project. And of course, I had really great help from my laboratory. I want to shout out two people in particular. So one is Rosalba Peroni. So she was a postdoc that worked with me and was very fundamental in developing this project. I also want to thank Judy Campisi, who is, her lab has really great world-class expertise in senescence cell biology. And there was a key postdoc, Abhijit Kale, who helped me with this project as well. And of course, a lot of other collaborators, including Charles Brenner, who did our mass spec, Brigitte Schilling, who was fundamental in some proteomic studies, our FlowCore director, Herb Kasler, as well as John Newman, Morten Schneiby-Knudsen, and Andrew Heckenbeck, who did a lot of our biostatistics analysis, as well as key collaborators, Dr. Ishihara, as well as Isam Ben-Sara, and of course, my funding from the University of California President's Postdoctoral Fellowship in the NIH. So with that, I'd like to thank everyone, and I also want to thank my current laboratory members at UCLA, who are really the future of my laboratory and a lot of the projects I discussed in the last slide. So with that, yeah, thank you again, and I look forward to the questions.